Institute of Alcohol Abuse, and so we're going to talk, of course, about alcohol.
All right, thank you so much, Rafael, for inviting me. This is a fun event. I hope we can get it very interactive. It's you've had two wonderful speakers in the morning, and then you've had lunch. I don't know which of this makes it the most tough for me to follow after this, but let's see if we can. We've on purpose lowered the temperature so that you'll be freezing so much you can't fall asleep.
Thank you.
Let's see. So I struggled to find a good title, but I think this is one that may to some extent actually reflect what I'm going to be talking about. So a translational perspective on the neurobiology and treatment of alcohol addiction. For those who don't know me, I'm basically a clinician. I'm a psychiatrist running a translational research program to develop novel treatments for alcohol addiction at the NIH. Although I will be changing hats in the near future, so this is the new place a year from now. But it's still going to be the same theme, which is translation from basic neuroscience of alcohol addiction and trying to make it useful for the patients, which is where I started and where I hope to end. And that is a very challenging task. So I put up as a little provocation for you guys. We've had a lot of wonderful basic neuroscience of addiction reviewed for us this morning, and that's great. The journey through understanding the basic neuroscience of addiction in animal models over the past 3 decades, I would say, is one of the most appealing intellectual journeys in science that I'm at least aware of. And so it's been fun to follow and to some extent be part of. But at the same time, as a clinician, there's a tremendous frustration because you need to appreciate that very little of those science advances have been translated into something that's actually useful for patients with addictive disorders. And so that's a dilemma. Along the way, we've gained a lot of understanding of basic motivational processes that are in operation in normal physiology, and a lot about the circuitry and the molecular events that underlie this, and yet the impact on the lives of people with addictive disorders is very limited. And in fact, it's gotten so bad. This is not unique to addiction. It's unique essentially to psychiatric neuroscience across the board. So my provocation to you is this paper by Steve Hyman, the former director of the Mental Health Institute at the NIH, who in 2012 put out this review paper in Science Translational Medicine, which basically says we haven't made any headway since the 50s when tricyclics were accidentally discovered. The reason is because we're stuck with animal models that are totally not predictive of anything clinically meaningful. We can't translate anything because we don't have any biomarkers that are neither diagnostic nor predictive of treatment outcome, and that is the reason why the pharmaceutical industry has rightly abandoned psychiatric neuroscience. We basically have to throw out everything we know and start over from scratch. Now, I don't necessarily subscribe to that view. I find it very extreme, but it's certainly an interesting challenge for us to sort of measure up our advances or lack thereof against. And so my task today that I've taken up on myself is to try to show you some things that actually might translate. And then, of course, we can, against the backdrop of those, think about things that perhaps we're not sure whether they do translate, and in some cases that are unlikely to translate. And I'll do it it will be very different. You know, this is first and foremost a clinician's perspective, and I'm still learning. Experimental psychologists are kind of intimidating to a physician because it's taking me all these years to figure out all the reinforcement schedules and all this stuff. I sort of have a hang of it, but not really. So this will be really driven from a clinical perspective, and and exemplified using a couple of journeys that we've made in our program through a few systems. You may think, I don't know if you still do, but too many years back to really give you the number, when I was contemplating what directions to take in neuroscience, I actually sort of looked at alcohol addiction and discarded it because at that point it didn't seem particularly interesting. It was, all the fun was in the cocaine field and perhaps opioids. Alcohol was this ill-defined substance that perhaps entered the lipid membranes of cells and somehow mysteriously affected things in a billion ways that we didn't quite understand. So I went off and started studying stress and emotionality. And of course, all these years later, and I'm still not telling you how many they are, it's gotten a lot more interesting. And so what I'll try to tell you today is that there are at least 3 reasons why even those of you who don't take a particular specific interest in your own science and studying alcohol addiction should pay attention. One is, of course, one that's lasting and remains, and that is that alcohol is, as you've already heard from Jeff Daly, one of the major public health problems that we're facing. But the other 2 are that it has very interesting, complicated, but very interesting pharmacology, some aspects of which I hope to show you today. And the third one is that it's illustrating in some very nice ways, beginning to illustrate principles of personalized medicine that I also will try to illustrate to you. So that's the 3 reasons. If you're not convinced, then I give up. I'm not David Nutt. No. David put alcohol at the top of the list of harm from drugs to society. But of course, the next thing that happened was he got fired by the British government. I don't want to get fired by the US government, so I just pass on numbers that the World Health Organization is putting out. This is Jürgen Rehm, of course, is the big name behind those analyses. But whether, no matter how we slice it, alcohol is among the top causes of morbidity and mortality in the world. So yes, nicotine addiction is, if you measure by the standard measure, the quality adjusted life years lost or other standard measures of public health impact, nicotine indeed does come out higher than alcohol. But that's kind of, I would claim that that's actually in a way an exception, because in the case of nicotine, it is not the drug that kills people. The drug is a vehicle that gets people to consume harmful substances such as tobacco, smoke. And in fact, we all know that nicotine itself can be safely administered. For some of my best friends write the best papers while enjoying the nicotine patch, and that kind of tells you that it's not really even though it is addictive in a similar sense, in many ways as other addictive drugs. It's a special case in the sense that the drug-associated harm is not caused by the drug itself. So if you look at the rest of the drugs where the harm actually is caused by the drug, alcohol really comes up on top. That's all I'll say about that. Well, actually it's not. So this is the translation of those numbers into the US reality. It's close to 100,000 people a year die from alcohol-related causes. And these people are typically relatively young when they die, so the expected life years that are lost are quite high. And through that, both the human and the economic impact is just tremendous. So there's a tension in this field between the people who do the public health work and the people who do the neuroscience, and I'll spend a few minutes trying to address that tension so you can come out better equipped to handle that tension, because you will hear a lot from people in the public health field that it's all about the total amount of alcohol that's consumed in a society. In fact, very influential people in the sort of that part of the alcohol field will tell you we should not look at individual factors, vulnerabilities, addiction as a disease concept and all that stuff, because all that matters are policy measures that reduce alcohol consumption in a society. And the most common argument that is brought forward to support that notion is that the amount of alcohol-induced harm is very closely correlated with the amount of total alcohol consumption in the society. So this is from the special issue of The Lancet a few years back, where Jürgen Rehm and others under the auspices of the World Health Organization mapped out consumption and then correlated it with harm. And so you can see, of course, this is the most scary example we have. This is Russia, and it is one of the few, developed countries maybe, where life expectancy is actually on a decline, and quite a bit of that decline is caused by alcohol use. And then you have, but closely sort of in the second tier, you have parts of of Europe, including Italy, and then you have North America. And so these are the high consumer countries, and the amount of alcohol-induced harm correlates closely with that. Before we dissect those numbers, just a little sideline, because perhaps with less well-informed audiences than this one, whenever I go out and talk about alcohol-related harm, people will say, well, what about those protective health effects of alcohol? We keep reading about those in the newspapers. And so you have to, again, be equipped to discuss those issues if you're in this field. And so this is just an illustration what that looks like. And this is now with a narrow view at cardiovascular morbidity. And you will see that even for that narrow perspective, there's a J-form curve, which means that yes, there are some relatively small beneficial effects. Those are observed at relatively low levels of consumption, and as you turn around and as you increase daily levels of consumption, you will very quickly end up in territory where the harm related to alcohol so sort of makes up for those benefits and plus some. Yes, Rafael?
But 20 grams per day is not so low.
Well, it's not fully two glasses of wine.
7 liters per year.
So I can't do the math in my head, but it's like, so the standard drink is about 12 grams, so it's not fully 2 glasses of wine. So so you have to remember one thing, though. This is just a narrow example of cardiovascular effects, right? Which is one of the few areas where you have any beneficial effects. And the other part of this is all that really matters in the end is all-cause mortality and morbidity, right? And so for other consequences of alcohol use, you don't even have this J, right? So once you pull it all together, you really have a very minuscule interval in which, yeah, maybe some small level of consumption is either harmless or beneficial, but it very quickly turns over into harm territory. Okay. So here's the dilemma, and here's the discussion and the disconnect between the public health people and the neuroscience people. If total alcohol consumption is what determines the public health impact of alcohol, why on earth should we be focusing on alcohol addiction? And the reason is this, and this is buried in ancient journals before PDFs were invented, so you can see that from my scanned copy, but this is a classic, right? So this is the Letterman distribution of alcohol consumption, which shows you that the vast majority of alcohol in a society is consumed by a relatively small minority of people. So as total consumption increases, it increases disproportionately in this minority, and this is where you find people with alcohol use disorders. So you're sort of getting an indirect measure of the exposure of those people. And so you have to, yeah, these people out here probably fine, but it's the people to the left in the distribution that, I'm sorry, it's the other way around. It's the people on the extremes of the distribution that really contribute most of the morbidity and mortality. So it is, even acknowledging the fact that total amount of consumption is an important factor, and even acknowledging that policy measures that reduce total amount of consumption are helpful, which is indisputable, good data to that effect, it still is meaningful to focus on the people with alcohol use disorders. And just to get this straight, looking at the DSM categories, we had, David was discussing those before, DSM-4 versus DSM-5. I would say that none of those are all that great. And so let's go over just a couple of thoughts around those. So, we used to have in DSM-4 this category of alcohol abuse, and a lot of times you'll hear the term abuse fairly generally. Please just drop it. In the sense that it's used sort of in the general conversation, it's very ill-defined. In the sense that it was defined in DSM-4, it just completely didn't make sense. By the time people that do epidemiology, such as Bridget Grant, who ran the NISARC study, started looking at what those, excuse me, it's the pleasures of being a government employee. Hope this won't be popping up every few minutes. If it does, you'll just have to live with it. So if you look at people who qualified for DSM-4 diagnosis of alcohol abuse, they were all over the spectrum of severity, and the vast majority of them got the diagnosis based on a single behavior. Can you guess what that is? What is it? Oh, come on. No, it's just a behavior, and it's, you know, so it's drunk driving. So people got a diagnosis. If you look at the criteria for alcohol abuse, people qualified for that diagnosis for most of them because they had been caught drunk driving, which already tells you that that's very much disconnected from any disease process, because in many cases that will be just, you know, about the norms where you live. In many cases it will be about discovery risk. It's very clear, you know, if you live in Sweden, it's pretty clear that if you're driving your car drunk around the city of Stockholm, your discovery risk will be much higher than if you're far. It's a totally meaningless diagnosis. And in that category, you would find people with no other problems, as well as people with fairly well advanced problems, probably neuroadaptations and all the stuff that we're going to be talking about in the neurobiological sense. So for those reasons, in DSM-5, the diagnosis of alcohol abuse has been dropped. What remains is a more unitary diagnosis of substance use disorders. Within that, alcohol use disorder. I don't like it because, as was pointed out, it lowers the threshold. But in fact, there are epidemiological studies that say that, you know, it's now grouped into mild, moderate, and severe. And the current DSM-5 category of alcohol use disorder corresponds to the old alcohol dependence in DSM-4 if you take it from about the moderate and up to the severe. The people with a mild disorder really were below the diagnostic threshold in DSM-4. There are some nice epidemiological studies from Wilson Compton and again Bridget Grant showing that for most addictions that mapping applies. But even so, I am not a believer in DSM categories, and I'll tell you why. A couple of reasons. The stability of these categories is not that great. There's a huge NIAAA sponsored genetic study called COGA, the Collaborative on Genetics of Alcoholism, and they had the tremendous opportunity of subjecting people to diagnostic interviews longitudinally. It was really amazing. You look at this is published years back in the Archives of General Psychiatry. You can go back and look it up. You take about 1,000 people and who had a diagnosis of alcohol dependence. And mind you, this is a lifetime diagnosis according to DSM-4 criteria, right? And you re-interview them 5 years later, and like half of them don't have a diagnosis anymore. Well, wait a second, it was a lifetime diagnosis. They couldn't all of a sudden lose their lifetime alcohol dependence. So point number one, the DSM categories are not particularly stable over time. Point number two, if you go out and do epidemiological studies and you diagnose people based on these categories, you will find out that only 1 in 10 of those people who qualify for an alcohol dependence diagnosis, which is the more severe syndrome, actually ever has received treatment. Now, one part of that is access to treatment is difficult. A lot of people who should have treatment don't get it. But there's also a huge population of people who are basically fine, even though they qualify for a formal DSM diagnosis and they go in and out of that diagnosis. So I've come to the conclusion that we need to listen to one more source of information. That was actually already pointed out in this classical paper by Griffith Edwards and his colleague Ross ages back, which says that the patient has something important to tell us as well, and the patients tell us something when they seek treatment. By the time you look at people who fill the diagnostic criteria for DSM alcohol dependence and seek treatment, you have actually a clinical population which has a tremendous predictive meaning in terms of consequences, in terms of prognosis. So when I speak about alcohol dependence, I will equate that with alcoholism or alcohol addiction, and I will refer to people who are in that category. well knowing that epidemiologically there's a lot of people out there who may fill the criteria and they're actually probably different, okay? So, yes?
Just to clarify, that
Yes.
mean that it's useful to have a close subset of people that seek the treatment? But what about those alcoholics that are really alcoholics that deteriorate in their life, social status, in family status, but clearly,
Absolutely.
and they don't care about treatment.
Oh, so it's this is the difference between sufficient and necessary, right? So I'm not saying that there are not people who are out there who would benefit from treatment and who share many important characteristics. And what I'm saying is that if someone fills the diagnostic criteria and seeks treatment, then I know they're in this category. There's a lot of other people in that category outside. It takes, on an average, even for those who do seek treatment, we know from those epidemiological studies, it takes on an average about 10 years from they fill criteria for diagnosis until they seek treatment, which is a tremendously long time. So you're absolutely right. It's not, a required element, but it tells us something that the people who actually do seek treatment are different from a lot of the people who fill the same diagnostic criteria and don't. Okay? So you could perhaps split it, if you'd like, into three populations. The people where we are probably over-diagnosing because our criteria have a very hard time capturing the key elements of a disease, and so we just over-diagnose a bunch of people where probably the key processes we're talking about are not in place. Then you have a bunch of people where they are in place, and those people are not, perhaps not yet, seeking treatment. And then you have the people who have their processes in place and do seek treatment. So it's easier, if you want to, for me, to focus on the people who have it, and those people have told us something very important, right?
I think the trouble is because in a stereotype way, and how movies books and all the pop culture portray alcoholism, but it's very difficult for them to admit that they are alcoholics. So for alcoholics to admit that they're alcoholics and seek treatment are really special.
Yeah, that's very interesting. So that's a different talk, and I could easily use up 2 hours for that talk. So that's largely a function of a lack of skills on the part of many people who try to talk to alcoholics. There's this widely held perception that addicts have resistance to treatment, confrontational approaches were all the vogue. And then once the behavioral sciences start looking at this in a scientific fashion, and we develop methodologies such as motivational interviewing and motivational enhancement, it turns out that phenomenon is more of a function of how you talk to people than a function of the people themselves. And by the time you learn the techniques to talk to people with addictive disorders, they have no problem admitting that they're in trouble. They just don't know what to do with it. And even when they know what to do with it, they're unable to stick with it because of many of the things you've already heard about. When you get this failure to admit that there's addiction is when you tell people you are an addict and I'm going to tell you what you need to do, you can bet that you're going to encounter a lot of resistance. If I'm sitting in my practice and someone comes to me and says, I have all this anxiety and my mood is low and I can't sleep properly, and I say, well, gee, here are some reasons that this could be caused by, and by the way, could we go over those, and by the way, what's your alcohol consumption? I get a very different response. So, as I said, totally different talk. There are techniques on that. It's amazing because the science has been in place for years and it's still not out there in actual practice. But if you look at the techniques of motivational enhancement and CBT, it's a totally different world from the confrontational approaches and the image that you just referred to. So that's actually a very good question, and thanks for bringing it up. No, addicts know for the most part they're in trouble. They just can't cope. Somehow, many of them just can't find the strategies to cope. Okay, chronic relapsing disorder. So, in some ways, it's again a tricky point to make. At one end of the spectrum, you have a perception that all addictions are the same. At the other end of the spectrum is this perception that every drug is unique, and as always, or as often is the case, the truth is somewhere in between. So in some ways, all addictions are very similar. So the chronic relapsing nature of alcohol addiction is very similar to that you see in other disorders. But at the same time, they're also different, as are other drugs. If you haven't read it, there's a beautiful review in Nature Reviews Neuroscience by Aldo Badiani, Yavin Shaham, and others, just even looking at differences between cocaine and stimulants on one hand and opioids on the other. Alcohol is probably even more different in many ways. But the real challenge is that even among people with alcohol addiction, they're not all alike. And we'll come to why that is. It's such a complex drug that this is really where you have the richest substrate for individual differences. So I've stolen this slide from Ken Kendler, my favorite psychiatric geneticist, and I've adapted with his permission. But nevertheless, he put this forward many years ago with regard to depression, but the same really applies to most complex behavioral disorders. And the thing is that you can look at the clinical phenotype. So this, here are people who fill diagnostic criteria for alcohol addiction, and if you want them to fill the Markus Heilig criterion of having stopped treatment, so be it. But there are people we're looking at, and they may look very similar with regard to many features that appear clinically very important and probably are. That doesn't negate the fact that they get to that point through very different trajectories. So you should think about this as an end-stage disease, and for the physicians here, if there are any, this is very much like congestive heart failure. It seems like a well-defined diagnosis, we know the symptoms, the legs swell and this and that happens, but the fact of the matter is we know that the pathophysiology that can lead up to this can be 100 different reasons, right? And the same thing goes here. So Kendler captured this nicely for depression. This is my reworking of that conceptualization for alcohol addiction. So we have this genetic susceptibility, about 50, probably closer to 70% heritability if you account for the error measurement, for the measurement error in the twin studies. But that doesn't really tell us a whole lot, because you will have some people who are strongly genetically predisposed, and Kendall calls them pre-kindled or already there, right? It takes very little additional environmental insult, including stress exposure and alcohol exposure, to get them to the disease zone. But others may have very little genetic susceptibility, and yet over many years of exposing the brain to alcohol and being stressed and being subjected to life adversity will end up in a seemingly similar phenotype. So our challenge is to understand the underlying heterogeneity, and the endophenotype concept has already been brought up today, and this is critical to everything I'm going to say from now on, because we need to deconstruct this complex disease phenotype into its endophenotypes. We need to understand the biology of those individual endophenotypes, and maybe many years later, once we've done that, we'll be able to reassemble this into different disease entities that will make more biological sense. And the reason we want to do that is because that will help us guide treatment choices. And I'll give you examples of how deconstructing this complex phenotype actually does help us match the right patient to the right subject. So I told you that the neurobiology of alcohol addiction is very complex. Many targets, the first wave of targets, and I don't know how much Rainer talked about this. I always try to sync our lectures, but Rainer usually talks about the first wave targets for alcohols being the ion channels, and then of course that being followed by a second wave of targets which are indirectly affected, among which you will find many of the systems that you're familiar with. So there's this tremendous richness of pharmacology that you can easily get lost in, and occasionally we do, but that also hopefully creates a very fruitful substrate for finding a multitude of treatment targets that ultimately, if we can parse out which pathophysiology is at play in which subject, we'll be able to hit with the right treatments. So, this is this lovely paper that I return to over and over again. Where should we look for targets in trying to develop treatments for alcohol addiction? And so you start looking at the circuitry, and this is all great, but as a clinician, I can't really cope with that. So this is my version. And it's really actually the same thing, because within each of these circles, you could map in the sub-modules that are in the big circuitry map, but it really is helpful from a clinical standpoint to try to group them into these three main groups of processes where we deal with systems that motivate approach behaviors, or you can also call them reward systems. You can look at the, so that was here, you can look at the systems that motivate, the stay away from dangerous stimuli behaviors, the aversion systems, with the amygdala, the nucleus ostrea terminalis, and dorsal raphe, and many other structures that are at play here. And then, of course, you have the relatively unique, or at least best developed in human systems of forethought and planning, and all the cognitive brakes on those subcortical systems that we use when we use our sophisticated cognitive control. And the thing is that alcohol targets all three of these big modules, so we could potentially look for targets in most of those. For now, we've been mostly focused on this, but I suspect that the richest ground for future treatments will be found here, and that's just an era that's opening up now. So, the first very silly, simplistic statement with, how similar, how different are drugs? The first question to ask yourself: we all know that drugs are addictive because they impact the mesolimbic dopaminergic systems, right? That's been on the official NIAAA website for I don't know how many years, so it's got to be true, right? Well, it's not necessarily the case. And you have to ask yourself empirically, what's the evidence that alcohol has an ability to activate these canonical brain reward systems in humans? So some indications in animals first. Yeah, alcohol can be rewarding. So this looks nice, right? Condition place preference. You can get reasonably robustly with alcohol, so surely it's rewarding. Camilla Carlson, who generated these data for another totally different paper, gets it to work every time. So it's great, except you have to know that you have to have exactly the right inbred mouse line. You have to have exactly one correct dose of alcohol, and it only works when Camilla does the experiments, because whenever we get a new student, it takes 3 months of the model not working, because it's so not really a strong pronounced effect compared to when you do CPP for cocaine or CPP for morphine or heroin. So alcohol can be rewarding by this classical measure, but it's much less robust than it is for classical drugs. Does it activate neurochemically the conventional brain reward systems? Well, again, the answer is kind of a bit of a mixed bag. This is a minor citation classic from Bert Weiss from ages back, when Bert was finally able to get microdialysis to work in animals that self-administer alcohol. And you will see that this looks very nice, right? Self-administration and dopamine levels in the accumbens go up. A couple of issues with that. First of all, this is a genetically selected alcohol-preferring rat, so that's great. If you go to a regular Wistar rat, you really have to pull out your magnifying glass to see the effect. And even if you look at these guys that are alcohol-preferring, the magnitude of the effect is about 1/10 of what you see with let's say, amphetamine. Right? So yes, it can, but it's sort of a different flavor. And then, and this is mice and rats, and if it's so different in different mice, different rats, you have to start asking yourself, what is it like in humans? And so there's not too many papers until fairly recently. There was a paper from Canada. Isabelle Beaulieu had a small paper with 8 subjects who got alcohol and had a PET session, and that indicated that there was some dopamine release going on after the alcohol administration in the ventral striatum of these people. And then Dan Homer and Jody Gilman in our program had this very nice paper which showed that by fMRI you can actually show an activation of the ventral striatum following a very tightly controlled brain challenge with alcohol. And the really nice feature of that paper was that the subjective feeling of intoxication correlated very strongly with the bulb activation of the ventral striatum. So nice, right? Alcohol is just like every other addictive substance. Not really. So first of all, there's a very interesting but also challenging sexual dimorphism in the dopamine response to alcohol, such that this is a paper from Nina Urban at Columbia University that really shows that in response to alcohol, men will relatively reliably release dopamine in the ventral striatum the way this experiment was run, and you will see the same kind of correlation between that release and the subjective intoxication that we saw using fMRI, but in women, this is totally absent. And in fact, in that paper, I don't say that I necessarily subscribe to that notion, although it's an interesting one. The authors put forward the hypothesis that maybe this contributes to the about 2-to-1 prevalence ratio of alcoholism that you have between men and women. Who knows? But
Did you
what?
correlate
Yes.
this with the phases of the menstrual cycle? Do we know?
I think they controlled for that.
They control for that?
Yeah. Yeah, it's a very nice paper. It was Biological Psychiatry a few years back. It's just very nice. It solved the dilemma for us that I'll come to in a little moment. So this is the next complication. So, okay, so I've shown you, yes, alcohol can activate these classical reward systems. It's sort of you have to qualify, and here you have to qualify further still, because we've had a series of papers from the lab. And the first that you saw, the one with a nice activation and a nice correlation, that was healthy social drinkers. And then Jody Gilman went on to study heavy social drinkers, some of whom actually had a diagnosis but had not sought treatment. And then in the last paper, we've actually found ethically acceptable ways to expose treatment-seeking heavy alcoholics to an alcohol challenge and look at the brain activity that results, and this whole activation just goes down. So you have a very, very linear progression of social drinkers getting a decent amount of activation and correlation between that activity and the subjective high, and then, as you would expect really, as you follow your trajectory into addiction, you get this allostatic shift, and basically now the drug is no longer activating this reward circuitry. So that's interesting. People are different, and one of the themes here is that when we say personalized medicine or tailored approaches to treatment, we typically think genetics, and I'll come to that in a moment, but people are also different in terms of their histories. And so when you want to develop a tailored or personalized medicine approach to treating people, you have to take in account not only the genetic differences, but also what kind of neuroadaptations are in place in that patient's brain, and how does that impact my ability to target a particular system with a medication, right? It's very clear that from these data, I hope that it's a very reasonable hypothesis, or actually statement, that if someone is in the early initiation stages of alcohol addiction and is responding with an activation of brain reward systems the way you've seen, it might make sense to treat them with a medication that will block those effects. But it's equally clear that if someone is as far into the addictive process as the patients where all this is gone, that's essentially meaningless, and in fact could make them worse by interfering with reward from natural reinforcers, and that is in fact probably the case. Okay, so I've told you that alcohol can activate the canonical brain reward systems, but with a number of qualifiers that I hope you'll remember. To the extent that that happens, it's an indirect effect, and much of that effect is driven by an alcohol-induced release of endogenous opioids. That has been a theory for many years based on animal experiments. As you probably know, there's an inhibitory influence on the VTA neurons by GABAergic interneurons in the VTA, and then there's an beta-endorphin input to that circuitry that can remove that tonic break. And so the idea has long been that when you drink alcohol, beta-endorphin will get released, and that will lead to a disinhibition of the dopaminergic neurons, and that's how the activation happens. The only problem with that has been that the translation has been lacking, and so therefore it was very much appreciated when Jenny Mitchell at the Gallo Center a few years back was able to use PET and C11 carfentanil displacement to show that in fact, following ingestion of alcohol, people would release endogenous opioids and thus displace the new opioid receptor tracer. So I always use this slide to sort of justify this cascade: alcohol, endogenous opioid release, disinhibition of mesolimbic dopaminergic neurons and rewarding properties, and of course that is cheating. And a sophisticated audience like this one can catch me, so please tell me how am I cheating in saying that this explains that? What's missing here? What are we looking at in terms of brain structures? We're looking at the accumbens and the orbitofrontal cortex, right? We're not looking at the VTA. It's big, so you know, I've been bugging them ever since they designed the study. You've got to see the VTA, and they can't because the scanner that they have access to at the Livermore Labs doesn't have the resolution to visualize this small area there. So there's still a little piece missing in this chain of evidence, right? Yes, in general, the statement has now been substantiated that alcohol is able to release endogenous opioids, most likely beta-endorphin in humans, but it still remains to be shown that really happens where we want it to happen, which is in the VTA, which would basically be summarized in this cascade that you know, endogenous opioid really, I already told you all of this, right? Okay, so this is such a great story because you've told people all this pathophysiology, and then it's just so nice and logical to come in and say, and therefore naltrexone, the opioid antagonist, is a great treatment for alcohol addiction, right? And this is the way we teach medical students. We tell them this is the physiology, this is the pathophysiology, here's the drug, it will fix the pathophysiology, and therefore people will get better. And of course, we never tell them that the way it really happens is we have no clue what's going on. Someone serendipitously discovers that a molecule will be beneficial clinically, and then we spend the next 15 years trying to figure out why that is, and in the process, we discover a lot of the pathophysiology. So that's how this really happened. But nevertheless, naltrexone is an approved treatment for alcoholism, and it's a very interesting phenomenon because it sort of provides the proof of concept for the idea that brain-targeting pharmacotherapy can be beneficial in alcoholism on one hand, so that's great. If people tell you, we're not sure alcohol really works, you should be able to rebut that and tell them, but I've heard that there's 50 randomized controlled trials out there plus some, and that there are meta-analyses that put together those trials, and by that standard measure of evidence-based medicine, naltrexone does work, period. And that's true. So you should really say all those things. But you should also know that very few people get an naltrexone prescription. Overall, the effect size in the meta-analysis is small. It's about, if those familiar with it, it's a Cohen's d of 0.2, which is in the range of what the good behavioral therapies are able to accomplish. But the most interesting and mind-provoking thing is that those of us who have been prescribing naltrexone from the early days, early 90s, quickly noticed that there's something weird with it, such that some patients seem to have this tremendous benefit, and that's a minority that we like to talk about, and I could spend a lot of time telling you all the anecdotes. And then we have a large population that don't seem to respond at all. And so you could write that off as random variation and the propensity of clinicians to tell the good anecdotes, and that's why you should never trust physicians with doing science. Or you could say, wait a second, the good clinical observation actually is at the root of many a good discovery. And if people in clinical practice seem to be responding differently to a medication, maybe that is because they actually are different. And of course, that brings us to the modern era of personalized medicine, where we try to figure out who might be responding to what, right? Of course, pharmacogenetics. And in this case, pharmacogenetics for all the usual good reasons, plus the reason that already in 1998, Mary Jean Creek's lab discovered that there's functional genetic variation in the gene that encodes the target for naltrexone, which is the new opioid receptor. And so you have a number of different single nucleotide polymorphisms, but you have one that's particularly interesting that encodes an amino acid exchange out here that's been extremely controversial. I've been stuck with this one for 15 years now, and finally we're beginning to piece the story together, so I'll tell you the story, the way it's coming together. For us, it started out, I should actually mention, it's very bad of me not to have a slide in between here, because obviously Chuck O'Brien has been at the forefront of this, and in the early days, Chuck made the claim that this functional variation would moderate the clinical efficacy of naltrexone, and they had a famous or infamous, depending on your inclination, paper in neuropsychopharmacology together with Dave Oslin, where they pulled DNA from several randomized controlled trials of naltrexone and made this claim, and people tore them apart, literally. And there were many weaknesses in that initial paper. We picked it up based sort of from more the biological and endophenotypic angle, and I think by now we're getting to the point where we can tell you the story and what's going on. So for us, this started by the observation that there's very similar variant in the rhesus macaque. So Greg Miller at the New England Primate Center published that around 2004, I believe. It's interesting in itself, evolutionarily, that even though these variants are different, they arose in these two primate species, namely ourselves and the rhesus, that share some features that you don't have in lower species, such as a highly organized social existence, and I'll tell you why that is likely to put some selection pressures on the evolution of this particular variant. But what we noticed, working with Christina Barr, who was still the fellow in the lab at the time, who's now faculty at our institute, we basically saw very early on that if we pulled monkeys with the functionally equivalent variant to that we find in humans, the minor allele, and we gave them alcohol, the males, but only the males, that carried the minor allele would respond with tremendously enhanced psychomotor stimulation, and yes, that did translate into increased motivation to consume alcohol and increased amounts of alcohol consumed. So that's published since years back, so I'm not showing all the data, I'm just making the statements. But it was really striking how strong the effect was, and it was very selective for psychomotor stimulation, didn't affect the other effects of alcohol, such as ataxia or sedation or anything else. So we basically went on to see if this translates into the human situation, and the pretty obvious hypothesis is that the cascade I've been telling you about, alcohol, beta endorphin release, disinhibition of dopamine, if that cascade is in hyperdrive in the minor allele carriers, in humans as well, then alcohol administration should result in a stronger dopamine release response in the ventral striatum of humans carrying that variant. So we basically carried out a raclopride displacement study using the D2D3 tracer raclopride to look at release of dopamine. I knew nothing about PET at the time, I probably still don't, but you know, if you're at the NIH, the nice thing about the environment is you really don't have to, because it's such a nicely integrated environment that you can walk into the office of the PET center director and say, hey, I have this interesting biological question, and before you know it, you're running a PET study and you're learning a lot about PET. So that's what we did, and to sum up, number of years of research in one slide, the results were pretty striking, because it turned out that only in the carriers of the minor allele did alcohol lead to any measurable release of dopamine at all. Now, we administer the alcohol in a somewhat unusual way. We bypass all the gustatory stimuli, with that all the conditioned cues that probably are also important. We give the alcohol IV to get a very tight pharmacokinetic control, because otherwise alcohol will vary a lot in blood alcohol concentrations and when the peak hits and so on. So we bypass all of that. Bidirectional Hamdani, who's the first author on that paper, has this really cool method where we can model the pharmacokinetics in any individual. We can tell the computer to take them from 0 alcohol, blood alcohol concentration to 0.08, which is the legal limit in most of the US, in 15 minutes, and we will get there within plus minus 3 or so %. And that allows us exquisite measurement precision, but at the expense of losing out on some of the indirect stuff. So when you do it like this, you see, and remember this is displacement of raclopride, or I know we still call it displacement, I guess it's more It's kind of embarrassing to say that when Diana is in the room because it's not really displacement, I know, but anyway, remember that the less of a binding potential that we detect after alcohol administration, the more supposedly endogenous dopamine has been released, and so you can see that in all the right places, in people carrying the minor G allele at this locus, you get dopamine release, and in the people who are homozygous for the major allele, nothing. So that's beautiful, I thought, but if you show that to a professional geneticist, they will tear you apart, because of course, that's one marker in this gene, right? And there's a ton of SNPs in that gene, and as you probably know, SNPs travel in packs, right? Remember, we have this one ancestral chromosome way back when in Africa, and then as we go through evolution, it breaks down into these haplotype blocks, and by now the size of a block is still 5 to 10 kilobases in a human, and so if you select people for one marker, there will be a ton of other markers that will follow, some of which you may not even know about. So that's the end of the road if you're studying humans. I mean, it's nice to show that image, but you still haven't nailed it causally as far as I'm concerned if you put on the geneticist's eyeglasses. So that's when you realize that translation is not a one-way street. A lot of times we think about translation as taking animal data and bringing them to the human situation, but equally often we benefit from taking a phenomenon that's not tractable in the human model and bringing it back to an animal model where we can break it apart into pieces, and that is just as valuable, in particular to nail down causality and pathophysiology. So in this case, and this is, remember, this was before the CRISPR technology was available, before a lot of the tools and toys that we have today to do these things. So this was quite a bit of work, but we made these mice that we basically humanized so that we ended up with 2 mouse lines, one of which essentially carries in the relevant parts of the sequence the human new receptor gene with the major allele sequence. And then in the other line, we have exactly the same sequence. We took it from the same guy, I know his name, but we did site-directed mutagenesis and we swapped out that one base pair in that one position. So you end up with 2 mouse lines that are identical throughout the 3 billion base pairs that they have, except for that one base pair, right? And you stick down, or you ask Larry Parsons at Scripps, in this case, to stick down a microdialysis probe into their ventral striatum, and you give them some alcohol, and you can see, you basically replicated that study. The guys that carry the minor allele release dopamine beautifully, the others barely do it, right? And now we think we can claim that this is causal, and in fact, this particular genetic variant is driving the differential activation of the dopamine response to alcohol. So that's great, but that's biochemistry. Does this translate into altered alcohol reward? So Elliot Robinson, who was in the lab and then moved to UNC and has kept collaborating with us, has addressed that question using the classical method of intracranial self-stimulation. That paper is just, I think, in the process of final revision for biological psychiatry, so you'll see it out soon. And so Elliot used ICSS. This is just his standard slide to show you that a rewarding substance such as cocaine will shift the ICSS threshold to the left, right? So enhanced reward, shift to the left. And here's the alcohol data, and so with the humanized new receptor mice, and so what you see here is that at baseline, there's no difference between the 2 lines. However, when you give them alcohol, the G line will lower their self-stimulation thresholds, the A line will not. And then this decrease that you see in the G line will be very nicely reversed using naltrexone, which of course won't reverse anything in the A line because there's nothing to reverse. And this predicts that the G line should be selectively sensitive to an opioid antagonist such as naltrexone, and in fact that is exactly what you see. So first you see here, this is kind of interesting because the association studies and sort of the effects of this polymorphism on alcohol intake have been all over the place, and in the mice we find that it's really variable. In some specific models, the G mice will drink more. In others, they will not. So this is 24-hour consumption in the home cage. They actually drink a little bit more. But if you do a 2-hour limited access measure, they don't. If you look at operant self-administration, depends on the schedule. But the one very robust finding that we've now seen by various measures, both operant and to bottle-free choice, is that the G mice will very nicely respond to treatment with naltrexone. And I'm not showing those data, but in the paper we also show this with nalmefene. And the A mice couldn't care less. Right? So you summarize all this, and it turns out that this provides probably the biology for the very controversial finding that in the meantime has accumulated enough of clinical data that people have been able to put together the very noisy individual clinical studies into meta-analysis. And by the time they did that, it does turn out that if you carry the minor allele at the neuropioid receptor locus, you have like an odds ratio of 2 or more to respond to naltrexone if you carry a G allele compared to if you don't. And as I said, by now I hope you've seen the biology. So this shows you just one aspect of where in some patients it will make sense to use opioid antagonists as a treatment, and we now think we understand the pathophysiology of that's behind the fact that these guys respond. But in other people, it will be totally meaningless, and if we keep diluting the treatment effects that are there in a minority of people in these large heterogeneous patient populations, we will miss treatment effects of future treatments, and we will underestimate the treatment effects of the treatments we already have. So this is the interim conclusions, and I already told you all of this. And so the question, yes?
What % of people of alcoholics carry that mutation?
Great question. So it varies by ancestry. So among Caucasians, it's about 15% allele frequency. So it's significant, but it's a minority. The really cool thing is that you have a wonderful gradient. So if you think about migration out of Africa, so African American, almost 0. Caucasians, about 15%. You go out to Orientals, who of course are protected from alcohol use by other factors, but if you look at the allele frequency, it's about 50%. And so there's a very interesting line of research coming out of UCLA, Naomi Eisenberger, and her trainee, I think his last name is Way, A-Y, looking at the behavioral genetics of this variant, and it matches up very nicely with stuff we've done in the monkeys. And to make it very long story short, it turns out that, this is an afterthought. Evolution didn't have any pressures through alcohol in this selection. Individuals with a minor allele are less pain sensitive, they're slightly more aggressive, they're more exploratory, and you can see the rest, right? So we were talking about, how can it be that some genes that seem, some gene variants that really seem to be non-adaptive today, that are retained in the gene pool? And so we can think about historical reasons for that, but it actually goes beyond that. There's a concept called balancing selection. There are essentially 2 kinds of selection. There's purifying selection. If a variant is thoroughly beneficial under all environmental circumstances, you will have purifying selection, and in the end, other alleles will be weeded out. But if you think about it, what's adaptive depends on what kind of environment you're in, right? If food is plentiful and there's little competition for it, then it's adaptive to stay in place, not wander off and take risks, and plow the ground and harvest the harvest. So it makes sense to have a number of traits retained in a population that make people do that. But when food is not plentiful, and especially in our evolutionary history, and you need to break up and seek out new areas to forage through, you need to have traits that are totally different. So this is captured by Bruce McEwen and one of his students in a beautiful paper that talks about hawks and doves, and how you basically, as a population, have an adaptive advantage for having both within the population, because that means that over time, as the environmental demands change, you will still be able to adapt to them through those different things. So that's very speculative, but there's actually some decent data to show that this is under selection, and it's sort of proportional to the distance migrated from our ancestral home in Africa. Yes?
There are some data also that show that the opioid receptor T specific SNP has an effect also on methamphetamine addiction. So do you think like naltrexone might have the same effect in other types of addiction, or is specific to alcohol?
So there's a paper, a mysterious paper in the American Journal of Psychiatry by Frank, well, I guess Frank is the last author, that shows beneficial effects of naltrexone in amphetamine addiction. And I guess we're all awaiting whether that finding will replicate or not. I keep an open mind. I don't pretend to understand it, but this could be a link. And that original finding, I mean, it's a very good study. But we should never trust any single study until it gets replicated. So we can pause, or I can take you into one more system, much more briefly, that shows a personalized medicine potential that we probably haven't thought about. It's totally up to you. And up to Raphael. Okay, so this is just a different take on stuff you've heard about already. This progression, it could just as well say from impulsivity to compulsivity, or from positive reinforcement to negative reinforcement. This is my former student, Wolfgang Sommer, now in Mannheim, Heidelberg, and his student, so I'm proud granddad, intellectually, I guess, their conceptualization of the same thing, but you've heard this over and over again, this progression, right? So we've so far talked about stuff that's probably mostly relevant for the early stages. Let's go into this thing. And along the way, you can induce an late stage post-dependent, as we call it, phenotype in rats by passively exposing the brains to alcohol. This used to generate a lot of debate because it was a dogma of the field that animals have to self-administer drugs in order to develop any of the features that are relevant for addiction. I've given up because all these years later, these guys it's very difficult to get a rat to do that with alcohol, just for a number of reasons that are technical. And by now, the Koob lab, our lab, many other labs have found that if you passively, at least with alcohol, if you passively expose the brains to sort of a pattern of concentrations and variation that's similar to what we think patients go through, you'll induce a number of the features that we thought would critically only arise if animals self-administer. So you see escalation, you see increased motivation as measured by progressive ratio. I'm not showing you, but you see compulsivity if you believe that that's what you see when animals engage in aversion-resistant consumption of drug. In this case, you can do it with quinine adulteration. So, we think that at least for a drug development screening model, it's good enough. Maybe there are some aspects that are not well captured. The useful thing about this approach is that this persists. This is a very persistent phenotype, so all this stuff is 3 weeks, or in fact could be just as well 3 months into abstinence. So this induces a very persistent phenotype. We know some of the molecular genetic underpinnings of those neuroadaptations. Gene expression changes occur in kinds of brain regions. One interesting behavioral phenomenon that occurs is that animals become sensitized to stress in this state. And it's kind of a two-part story. One of it is that stress responses become enhanced, but the other one is that this history of dependence seems to set up a connection between stress and motivation to consume alcohol. So what you see here is animals that have a history of dependence or don't, so you see the escalation, and then you expose them to stress, and without a history of dependence, it's actually very hard to increase alcohol consumption in rats under these conditions using stress. But if you look at the dependent animals, that goes very well. We have some fMRI data to show essentially the same thing in humans. You show them nasty IOPS pictures, and you look at the circuitry that responds to aversive stimuli, the amygdala, the insula, and the activation is much higher in people with alcohol dependence than in healthy volunteers. So there's a number of systems that we think undergo neuroadaptations as people and animal traverse through this process, some of which, stuff I will talk about other occasions, but for now we've talked about this, and this is a within systems adaptation. There's this 87 or 88 paper by George Koob and Floyd Bloom in Science that I keep returning to, and they introduced this concept of within systems and between systems adaptations. It's actually still a very useful conceptualization to have. So a lot of the stuff we're looking at so far is within systems adaptations, and then these other systems kick in, and that's systems that get recruited in that are between systems adaptations. So today is the within systems day. So we talked dopamine. Here's glutamate. It's kind of interesting. It's target for, supposedly recruit the hyperglutamatergic state in alcoholism. Supposedly that leads to escalation, and supposedly that's target for the clinically approved treatment acamprosate. You can see here acamprosate, again, personalized medicine for rats. It will only affect the rats that have a history of dependence and have escalated. In fact, it doesn't do anything to the rats that didn't have a history of dependence and didn't escalate. And I can show you the translation of that using MRS, because in fact, if you measure the glutamate levels, at least in the anterior cingulate of alcoholics, and you treat them with acamprosate or placebo, you will see that in placebo-treated people who are admitted to a detox unit, you'll have this progressive rise in the glutamate levels. If you have started them on treatment with acamprosate already here, instead you will actually dampen this hyperglutamatergic state. So that's already, it's right here, again, personalized medicine, right? But now it's not genetics, now it's having the history of dependence that pushed the glutamatergic system. Last one. So I'm embarrassed to talk about endocannabinoids with Raphael in the room. I don't know anything about this system. What's that? Yes. Yes, of course.
Getting back to the previous slide. Yesterday also we talked, actually we heard about this.
Mm-hmm.
And then at the end we got it that the calcium was mainly right.
I know, yeah, that's got to be Reiner. Hey, yeah, what can I say? I got the questionable pleasure of writing a commentary for Neuropsychopharm when that paper was coming out, and I didn't quite know how to turn. Look, the data are the data. It's a very provocative idea. Actually, in the end, it doesn't really matter from my conceptualization whether acamprosate serves as a vehicle for delivering calcium, and the calcium is what brings down the glutamate levels somehow. That's fine too. However, I would say that for now, this is a hypothesis, and we would have to have a lot of additional data to say that that's the final truth. So I keep an open mind. Reiner is one of the most solid guys ever. He didn't believe in this himself. They went through years of work before he said, I think it's going to be calcium. And so I respect that. At the same time, it's obviously such a provocative notion that, once again, it needs more independent replication, and in many other things, we should always be very critical, even of our best friends and of our own data. That's all I can say. But for the concept of hyperglutamatergic state, and that being sort of present selectively in people with a history of severe dependence, and therefore those people being selectively sensitive to drugs such as acamprosate or maybe an mGluR2 positive allosteric modulator that are able to bring that back down, that doesn't change the fundamental notion that you need to find the right patient, right? So as I said, I don't know anything about this, but I've noticed that there was a whole movement in the field that said that alcohol is producing a lot of its reward by activating endocannabinoids. And in fact, if you give people blockers of the CB1 receptor, that will block those rewarding properties, and therefore rimonabant is going to be the treatment for alcoholism. And some of that stuff, this is just some semi-randomly picked papers, one by George Kunosh at the NIAAA, showing mouse data in support of this. We had some little exercise with that as well. We potentiated endocannabinoid transmission specifically in the prefrontal cortex. You basically convert non-preferring rats to preferring rats. So we were on board with this. We just thought there were so many good people in that area that it was pointless for me to try to understand it. beyond that. So we just waited for rimonabant to become this wonderful treatment, and then of course it didn't quite play out that way. And in addition to making people miserable, anxious, and suicidal in some cases to the point of being pulled off the market for obesity, rimonabant turned out to be not efficacious for alcohol addiction. So, you know, people forgot about that and moved on to the next thing. But there's been a resurgence of interest in the interplay between alcohol and the endocannabinoid systems, and this is in the context of stress regulation. So I told you about this recruitment of stress sensitivity that happens as people progress into their alcohol addiction, right? So, slew of papers recently indicating that endocannabinoids, and most likely anandamide itself, and most likely within the amygdala, is a major endogenous buffer, not in the sense that it will sort of, at a basal, steady level, make an individual less fearful, anxious, what have you, but once the fear responses are activated by a stressor, activation of anandamide in its activity at CB1 receptors will be a major mechanism that the body mobilizes to turn that response off again. And it's involved in a ton of other stress-related things, such as fear extinction and stuff, but you know, beautiful work from Andrew Holmes. Well, anyway, but I'll just stick with this somewhat simple notion of being a stress buffer. So in that context, we got a chance to work with Bob Innes's pet group at the NIH. The nice thing of being a neighbor of Bob's is that he occasionally develops novel pet ligands that he will actually let us use. Not always, sometimes. And so Jussi Hirvonen was a fellow between our labs, took that new CB1 ligand and worked with us to look at CB1 receptor densities in alcoholics, treatment-seeking heavy alcoholics on our unit. Very striking results. You know, these are areas of differential binding potential between alcoholics and normals, and you see this widespread reduction in CB1 densities. And if you just look at one, yes.
So what was the ligand?
So the ligand is this one, F18 FNPEP-D2. If you look at one specific region, that's just a quantification, but it actually looks pretty similar almost throughout the brain. So kind of interesting, the amount of reduction very nicely correlated with the duration of alcohol use. So here's one sign that this is a reflection of a long-term neuroadaptation that occurs as people make their trajectories through developing alcohol addiction. You know, obviously this would be confounded by age, so we parsed out that age is not the reason, it is in fact the duration of alcohol use. And this doesn't reverse. So at least we scanned these people twice with almost a month in between, and there's, yes?
Did you get the same result post-mortem?
So by now I can tell you yes, but we didn't know that at the time. There's another result post-mortem that I'll show you in a second that very nicely validates this, which was actually a surprise.
Can you correlate with the emotion? You know that depressive patients have a very nice regulation of the C1 receptors. So have you correlated with the possible depression status of these patients?
Not really, not really. These are mountains of data. We probably still have some analysis left to carry out. At this level, at this point, we were mostly interested in a couple of other things. Let me show you what those are. But let me also mention to you that there was a parallel paper where Innes's group collaborated with Marilyn Custis at NIDA to look at cannabis users. And as you would more easily predict there, they're downregulated in CB1 receptors as well. That downregulation is more profound, not so strange, it's direct agonist, but that downregulation normalizes within a month. This is less pronounced, but it doesn't normalize. So there's something broken in these guys.
Are they polydrug abuser? I mean, does it take
Do they smoke? Do you factor for these things?
So these guys are relatively clean for a modern day alcoholic in an urban area. They're not totally clean. We've carried out all kinds of sensitivity analysis for smoking in particular, because that's hard to get rid of, and that doesn't seem to be the reason. So here's the nice validation of the PET method. And also sort of coming in through the back door is your pharmacogenetics and personalized medicine. So here's an intersection of history and genes. So there was a very nice paper in the Archives of General Psychiatry with a postmortem analysis where Ken Hutchison and his group look at the role of a functional genetic variant in the CB1 gene, CNR1, for Bmax, binding capacity. And you could clearly see that in individuals with one of the variants, you would have a higher receptor density than in the other. Well, here's the same people, and this is now independent of the history. So in the healthy volunteers, you very nicely replicate this, and this is still there in the alcoholics. And this is with a very small sample size. Again, you can see that the combination of prospective genotyping and PET with its excellent measurement properties allows you to find genetic effects in relatively limited population samples. And here, I showed this to Ken Kendler, and Ken is, of course, the man behind the Psychiatric Genetics Consortium. He doesn't believe in anything that's been found in less than 10,000 people. And I showed him, Ken, look, 20 people of each, and said, can't be true, needs to be replicated. So I pulled out this paper and told him, in fact, this is the replication. And he said, well, I guess we better believe it. So it's nice, it validates the approach. But the really nice thing is, so now you have to, so here's your intellectual exercise for the day. So we have seemingly two processes going on at the same time, right? Because there are some association studies that indicate that having this allele that endows people with a higher density of receptors will somewhat, not a whole lot, but somewhat increase the risk of developing alcohol dependence or alcohol addiction. And yet at the same time, I'm telling you that the people with severe alcohol addiction have lower receptor densities. What's going on here? Well, what's going on is what I've already told you about. What gets you in trouble is not necessarily what keeps you in trouble. We need to look at initiation factors as one thing, and neuroadaptations that occur over the history of developing addiction as something else. And here, this is allostasis, just, very nice case where people may, to some extent, be prone to consume alcohol because alcohol reward will be enhanced initially when they drink because they have elevated levels of the CB1 receptor. But as they do this, they undergo this allostatic shift, and so ultimately they become less sensitive to both to rewarding properties of alcohol, and they will become less effective at coping with stress reactions, because I told you that this is a system that people need to buffer the stress responses. Here's the illustration of, well, I'll just skip this. So here's a little experiment of nature's, again using genetics as a probe. So anandamide is mainly broken down by fatty acid amide hydrolase, FAAH. We think that it's this buffer on stress responses, right? Well, if there's functional genetic variation in the gene that encodes the enzyme that breaks down anandamide, that leads to a less effective degradation, wouldn't you think that those people would be better at buffering their stress responses? So we looked, and there's this variant that seems to produce a less stable FAAH, and so it's essentially like inhibiting the FAAH. And those people underwent the stress challenge, and you can see that these guys, so this is a guided imagery stress challenge using Rajita Sinha's, you basically carry out an interview with someone about a very stressful experience they've had, and then you construct a 5-minute auditory script, and you present that back to them, and people respond with very robust anxiety responses. And the magnitude of the initial response is not different between the people with the two different variants, but the people who are sort of naturally FAAH inhibited turn the response off much more efficiently, right? So from that arises the notion that if the endocannabinoid system has undergone these long-term neuroadaptations that have rendered it less efficient, the system will contribute to people being prone to seek alcohol for its negatively reinforcing properties, right? And if we can restore some of that functionality by blocking FAAH, we may actually remove an incentive for alcohol seeking and consumption. So it's totally opposite to the naive original notion we had that blocking the CB1 receptor would be a treatment. Now we're actually looking for a way of potentiating endocannabinoid signaling, and we're actually starting up a study with an FAAH inhibitor in September. So, a couple of years from now, I'll be able to tell you if this bears out. So that's the last thing I have. And so I just told you all these things. The endocannabinoid system is complex because it's important both in the early stages for alcohol reward, and then in the positively reinforcing incentives for alcohol seeking, but it's also important late for stress reactivity. And so you can see that by the time people become treatment seeking and have all these neuroadaptations in place, it may actually make sense to revisit the endocannabinoid system as a treatment target, and that an FAAH inhibitor might to some extent restore that functionality. And there's a ton of other stuff, and basically my take-home message that I would like you to get is that for each of these systems, we're more, it's not like the new receptor is a unique case and it was fun to play around with its pharmacogenetics, but that is the end of it. It seems that almost any system we look at, we need to consider that people are different, that there's likely to be functional genetic variation, and that functional genetic variation combined with the individual history will determine who will respond to which intervention. And so it makes life a lot harder for drug development. It's in fact a nightmare, but it also makes it a lot more interesting. And with that, I'll stop and just show you all the people it takes to do this stuff and take any questions.
So that variant that you showed for the fly, is that the same that Bentravat reported on with street drug use and alcohol?
Yes, 335. Yep. So there's an association signal there. That's been all, again, so it shows you the value of the alternative approach that uses endophenotypes and uses prospective genotyping. Association studies are a nightmare in terms of power. Groups are heterogeneous, phenotypes are heterogeneous. The math that you're using, which is essentially extended versions of the chi-square test, logistic regression is just that. So your power is miserable, and so sometimes we see a signal, then the next person can't replicate it, and that's where we come away with the notion that, nothing in psychiatric genetics replicates. The moment you turn this approach on its head and you say, I'm going to take potentially biologically interesting variants, I'm going to pre-select people based on those variants, and I'm going to try to find an endophenotype that's as close to the biology as I can make it, and measure it under as tightly controlled conditions as I can, the whole game changes. So anyone who wants to, so sort of the paradigmatic paper for this, that you guys should go back and look up if you're interested in this, is the paper by, we didn't invent this. Ahmad Hariri, working with Danny Weinberger, had this beautiful, beautiful paper in Science in 2002, I believe, or so, thereabout, with a serotonin transporter, long-short promoter length polymorphism. It was as controversial as any of this stuff. People kept debating. You had Peter Lesh saying it makes people more prone to being anxious, and others were saying, no, we can't find it. And then Ahmad recruited two groups, each, I think, fewer of 15 per genotype, put them in the magnet, showed them scary faces, and looked at amygdala activation, and you had twice the activity in the people who were short compared to the people who were long. And everybody said this can't be true because the group sizes of like this cannot find a genetic effect. And so a number of years later, there's a very nice meta-analysis, I think in biological psychiatry, that Ahmad and others did, and it's rock solid. The effect size is a bit smaller than they thought initially, but it's there. So that's the paradigmatic paper, and there has since been small but significant numbers of approaches like this that have been tremendously successful with the serotonin transporter, with COMT, and now with the mu receptor and a few others. Yep. Are people totally exhausted? Yes? Oh no, help. Now it's going to get difficult.
Completely unrelated to imaging. So wouldn't this sort of call into question exactly the way we do clinical trials? I mean, yesterday we were talking about the huge placebo response you have in psychiatric clinical trials. One of the things that had struck me about naltrexone for alcohol dependence is that the number to treat seemed to keep going up with the number of clinical trials that were done. And now what you're demonstrating is that there's an explanation for that, is that the more heterogeneous you make a population, the greater the number to treat becomes. So wouldn't we be missing a number of potentially viable treatments by performing clinical trials as has historically been done?
Yes, thank you for making that point. Absolutely. And in fact, I mean, the short answer is yes. And let me give, the annoying thing is, we've known this for decades. What's the effect size of imipramine? Anybody here old enough to know even what imipramine is? Tricyclic antidepressant, right? So what's the effect size? I don't know the number. The right answer is huge. If you look at the original studies, where they treated a relatively homogeneous populations of severely depressed people who were hospitalized for the depression. And then, as the SSRI market exploded and became profitable, everybody wanted a Me Too drug. So what do you do if you have tons of drug candidates that are seeking their place in the market? You have to run lots of studies to get lots of subjects, so you go out and advertise and you pay people to participate in studies. By that time, the severity of people in those studies, which is one reason for heterogeneity, was half or less by Hamilton Depression Rating Scores compared to the people in the original studies. Guess what? There's a meta-analysis that shows that the effect size of antidepressants, both tricyclics and SSRIs, varies in a very orderly fashion as a function of the baseline severity of depression. So we've known this, and still we have to repeat the mistakes of old days, and I'm sure that a number of potentially useful antidepressants have been missed because of this, and there's a risk that the same thing might happen in alcoholism trials. Just to reinforce this, I forgot to mention, there's a very nice paper, I think, again, American Journal of Psychiatry by Hank Kranzler and others, showing that you have yet another drug, topiramate, which again shows pharmacogenetic variation. And it seems that even though that effect size was actually okay, people were quite impressed by it, it's carried by a subpopulation, which I think is about 40% of people in that case, with a specific genetic variant of one of the glutamate receptors. Okay? And if you don't have that variant, don't bother taking topiramate. It's totally flat. I mean, they don't respond at all. Now, again, awaits replication, awaits mechanistic studies. Each of these cases will take a lot of work to tease apart the way we've been fortunate enough to be able to do with the mu receptor. But the reason I keep retelling the mu receptor story is because it sort of serves as a paradigmatic example, and it's nice to have all the steps from a humanized mouse all the way to the clinical meta-analysis. Yeah. But you know, we tell this to people in pharma, and the only consequence is they say, oh, it's even more impossible to run clinical development for medications for alcoholism than we already thought. And we already got out because of the field because we thought it was too hard. So it's kind of a double-edged sword, and I think academia and academic scientists will have to step up and fill that void. And I think the way we can do it is, we will never be able to run the definitive clinical trials, but if we figure out the endophenotypes and the biomarkers that will help define the populations, and we can sort of turn over that knowledge to the drug companies, then it will again be possible to engage them, and then we will be able to facilitate rather than scare them off from drug development. Have I preached enough? Yes?
The slightly switching gears, because you talked a lot about pharmacology of addiction treatment. Are there any emerging
wonderful question, thank you. So, my highly personal, highly speculative, and highly provocative take on this is we are at the end of the road. We've spent decades developing behavioral treatments, and actually, some of those are pretty good. So as I already mentioned, motivational interviewing / motivational enhancement, CBT-based relapse prevention approaches, contingency management, all these have a solid evidence base, and the real problem is not that we don't have behavioral treatments or that we don't invest in research on those. The real problem is people are not getting those treatments. People are instead asked to pay $40,000 for 28 days of not getting these treatments, right? However, having spent over several decades, both NIDA and NIAAA spent tens of millions, if not hundreds of millions of dollars on developing behavioral therapies. I personally have come to believe that, good though they are, that you sort of reach an asymptotic point, right? And it looks like it's hard to push it beyond what we already have. So I think the next generation is pharmacotherapy and combinations of pharmacotherapy and behavioral therapies. And I think at this point, there's little willingness to fund additional behavioral treatment research. The other part of your question that's actually even more interesting is, could there be genetic moderators of who responds to what in terms of behavioral treatments? And it sounds kind of like, space science, but in fact there are some indications from other parts of psychiatric neuroscience, namely treatment of social phobia, I believe, that indicate that there might be that kind of effect. So there's an interaction between people's serotonin transporter genotype and how they respond to standard cognitive behavioral therapy for their phobia. Those studies are a nightmare to carry out because they're extremely noisy. Anything that's behavior, so it would require probably thousands of people to figure out, so it's even harder than the pharmacogenomic studies. I don't see those studies happening anytime soon, or maybe ever. I mean, it's conceptually very interesting. I'd love to know, but it's just too hard. Yep. Yes?
What do you think about varenicline or other nicotinic receptor agents for alcoholism?
Well, so the initial animal data from Salim and Bartlett were clearly very compelling. As you probably know, there was an NIAAA funded 5-center study that recently got published that seems to support that those findings translate to the human situation. At the same time, there's a couple of studies, some of which are not published yet, one of them in Sweden by the Swedish Clinical Trials Network, that were negative. So
did any of them control for genotype at alpha 5?
No. And we have the DNA from Ray Litton's study in my lab, and we're genotyping away as we speak. And so a few weeks from now, I hope to tell you if that's a moderator of the treatment response. Yeah, that's absolute. So that'd be fascinating. There could be other, but there are other differences as well. So one should, this gets technical in terms of clinical trials design, but actually Ray and the NSIC collaborative that he runs have figured out a phenomenally smart way of running these studies. So the placebo response plagues every study in psychiatry, as Diana already alluded to. They figured out ways, based on the many studies that the NIAAA has funded before, including the combined study, of what are the characteristics of the person who doesn't respond with a placebo response. So they kick them out. I mean, the people who don't have those. So one characteristic I would never have thought about is lots of people lower the drinking dramatically already the moment they sign up to participate in a clinical trial. And those people never respond to a drug. So one simple thing is, if you decrease your drinking in the period between screening and randomization by a certain amount, goodbye. And so if the Swedes reanalyzed their study, based on those criteria, okay, it's post hoc, but they end up with a significant result. So it may just be a technicality. So we need to keep an open mind. It's early days. We need independent replications, but I think it's looking reasonably promising, and I would be fascinating to find out if there's a pharmacogenetic moderator of predictor of response there as well. That'd be great. And I have to. So aren't you worried? I mean, every time I tell these things to a clinical audience, they look at me and say, "Well, that's all great, but your average GP won't likely have a PCR in his back office anytime soon. So what's the use of any of this?" What's the answer to that?
We're under $1,000 genome already.
I'm sorry?
We're under $1,000 genome. We're all going to
Thank
end
you.
up sequenced when we're born.
You only do it once in a lifetime. And it's not even an issue of your GP doing it or ordering it. It's an issue of you doing it once and you get your genome. It's not someone else. And preferably you get it on a card or even better on a chip that's implanted sub-Q. And you go to your doc. I'm telling you, this is 15 years from now or 20 years from now, this is bound to be the reality of clinical practice. You go to your doc and you are diagnosed with something, they contemplate a number of treatments, and they'll say, and by the way, in trying to determine whether you might be harmed by one of these and what your chances are of being helped, might I access your sequence? And either the sequence will be in the cloud or on that chip, and if you choose to say yes, they'll run your profile against what's known about the medication, and that's how it's going to eliminate the need for any expensive additional genetic analysis. And by then, a genome will be $100.
Try to convince the pharmaceutical companies about that. That's the problem.
So that's going to be the bigger challenge. How do pharmaceutical companies make their drugs profitable when you can no longer hope for blockbusters, but instead have to look for niche drugs? And that's a very difficult discussion. One possible answer to that is, well, guys, if you want to survive, you'd better harvest whatever profits you can because you haven't been doing so great lately, right? So it may be that ultimately the pharmaceutical companies are forced into accepting this reality, or maybe not. I don't know. That's more of a business model decision. Yeah.
So, go ahead.
You-
Go, please.
Please, no.
I was intrigued. What are your thoughts about, you seem to repeated the message that NIAAA is saying, that addiction is like any other chronic disease, diabetes and others, and obviously there's a huge element of chronicity. But based on what you said now, all the discussion, and then the statement by NIAAA, and we all repeat, do you think those two go together, or are we shooting ourselves in the foot by saying these things, or?
No, I think I think quite the contrary. I think we still haven't fully taken in the implication of that realization. So the paper from Tom McClellan and the guys is 14 years old now, and most of addiction medicine is still practiced as if it never were published. People continue to view the failure to cure a patient as a reason to not provide treatment at all, when in fact what's needed is to adjust expectations and find chronic disease management models much the way any sensible diabetes nurse has figured out ages ago. Now, it doesn't mean that there aren't some people in whom all these neuroadaptive changes and all the social changes that happen might reverse, but certainly for the vast majority they don't. Now, I dream of, look, so if you ask me about my dream, so far we're correcting, or trying, in developing treatments, we're trying to correct for the maladaptive changes, neuroadaptations that occur and somehow get stuck, right? Now, a dream is to think maybe there are some switches somewhere which get pushed as this process moves forward, and maybe if we figure out what those switches are, we can find out ways of flipping them back to the pre-morbid state, right? And to be much more concrete, if what, one set of switches is that your DNA gets hypermethylated in the medial prefrontal cortex, turning down the expression of a number of critical genes that are required for that structure to exert its inhibitory control. Maybe if we give people a DNMT inhibitor, we can erase those methyl marks and restore that control, in which case the statement would no longer be true that these people are for, that's science fiction. I dream to see it realized, but I also am fully aware that it may not be feasible, and part of it is a lot of the changes may not even be at the level of regulation. They may be structural, and those may not ever be accessible for that kind of approach. So I don't know. For a lot of people today, the reality is very clearly that addiction is a chronic relapsing disorder. It is still a reality that most of the addiction medicine field has not adapted its provision of treatment to that insight, and we're all the worse for it. That was hopefully a very passionate plea, because I hate to see people who are already at disadvantage get a second mortgage on the house, go off to the Betty Ford Center, pay $50K, come back with this dream of a cure, or even worse, they go to some really nasty place like the Palisades in Malibu when they pay $80,000, and then they relapse, and by then they're failures, and it's something wrong with them. And it occurs to few treatment providers to say, well, maybe it's something wrong with us. And so that still really needs to influence our field better than it has. Yeah. And you had one more.
Yes. I want to know about the strategy for using other alternative in vivo.
Yep.
Like this one?
Yeah, say so, Frank.
How is the strategy exactly for using research? After treating the addicted person, or during the treatment?
So disulfiram is anti-abuse, right? It leads to this terrible accumulation of acetaldehyde, makes people flush, develop tachycardia, become nauseated, and actually can be very dangerous if you drink and if you have a heart condition. So the idea is that the risk of those terribly unpleasant consequences should deter people from drinking alcohol. Guess what? Addicts are not stupid. They realize, and because this is how it's supposed to work, it doesn't do anything for the cravings and for the negative emotional state and all the stuff that maintains drug seeking. So once they become miserable enough, they just discontinue treatment and they relapse. So meta-analysis of unsupervised anti-abuse treatment very clearly show that it has no effect whatsoever. It's a drug that's been on the market since the 50s, I believe, and used the way it's most commonly used. I told you how it's supposed to work, and then I told you how it doesn't work. Now, it doesn't mean it doesn't have any place in clinical practice. A lot of times you'll have populations, just like with naltrexone for heroin addiction, for instance, you have populations that are highly motivated, such as a physician who's on, so I ran a clinic like that at the Karolinska. I had a bunch of docs who would lose their license unless they came and under supervision received anti-abuse 3 times a week, which essentially covers the whole week. And they stick with it because, it's I guess it's a discrete choice procedure, right? Take your anti-abuse, lose your license. So it has a place in clinical practice, but not a big place. There's another aspect of disulfiram that you may be or may not be aware of that's very different. It sort of had a resurgence recently because there was some inexplicable data that disulfiram in animals would be beneficial for cocaine to reduce rates of cocaine self-administration. And there were some uncontrolled clinical studies that supported that notion, and then the whole thing got stuck with the FDA for some really silly reasons. So I heard from people who went to the IND meeting that they were asked to first run an alcohol interaction study with disulfiram in order to get to run a trial for cocaine addiction. And if you think about how silly that is, so it never went anywhere. But there are now so it turns out a lot of people thought that the effects, beneficial effects or seemingly beneficial effects, would be simply because it reduces drinking and then it's easier for people to abstain from cocaine, and so maybe that's, but it turns out that's not the case, and it turns out that disulfiram and a number of other enzyme inhibitors are able to inhibit a very unique enzyme in the synthetic pathway of dopamine. And so Ivan Diamond has, and Gilead Pharmaceuticals have now a compound that does nothing to inhibit the breakdown of acetaldehyde, but still has that function to inhibit the effects on dopamine synthesis, and that molecule is actually in development. But so that's a totally different mechanism. There's a beautiful paper in Nature Medicine, if you look it up, Ivan Diamond, Nature Medicine, probably 5 years ago or so, and it's now in clinical trials. Yep, with that.
Looking at the efficacy in the case of cocaine dependence, we still think that the effect on dopamine beta-hydroxylase would be important not only for cocaine but also probably for alcohol.
It could. Although the affinities are such that you get an accumulation of acetaldehyde probably before you really get pharmacodynamically relevant effects, dopamine beta-hydroxylase. I don't know. That's a tough one. It's quantitative. But it's a nasty drug. It's just too dangerous. I really wouldn't. I wouldn't recommend it. But some interesting mechanistic insights may still come out of it. Okay, are we done? Thank you all. Great question.